Welcome to my cell. So here we have the nucleus. And inside the nucleus you have molecules of DNA, which are much longer than this, but this is a sample of DNA. Also inside the nucleus there are these little nucleotides floating around. And there are two different kinds of nucleotides. There are RNA nucleotides, which have a phosphate, a ribose sugar, and a nitrogen base. So you can see phosphate, ribose, nitrogen base. And then we have DNA nucleotides, which are a phosphate and a deoxyribose sugar and a nitrogen base. So the nucleotide consists of those three parts. And as I said, we have two types of nucleotides. Now, at this moment, we are going to make a protein. So to make a protein, our first step is we need to unzip the DNA. And the enzyme called helicase is the one that does the unzipping. So we have the helicase, works its way up and unzips the DNA molecule. And it does so only for the section of DNA that contains the code that we are trying to copy. So step one is to start to build using the enzyme RNA polymerase. RNA polymerase will read the sequence of nucleotides on this template strand of DNA. So T goes with A. Now we have to use an RNA nucleotide that has the ribose because we are building RNA. So then next you go with the U because RNA does not have any thymine. Instead it has uracil. And then next it will be G, but this one doesn't work. RNA polymerase will not choose this nucleotide because it has deoxyribose instead of ribose. So get rid of that sucker and find one with ribose. So now I'm just going to keep building. And RNA polymerase would be bonding this ribose to this phosphate and this ribose to this phosphate to bond them all together. And because I don't have the ability to do so, I'm using this little strip with Velcro. So just pretend this strip doesn't exist. And now I am going to continue to build. There we go. We have built a chain of RNA nucleotides using the DNA as a template. This process is called transcription. Now that we have the code for how to make a protein, this mRNA can leave the nucleus. Okay? It'll leave the nucleus and go over here to a ribosome. And then the DNA will wind right back up and zip up. Now we've reached the ribosome, and the ribosome will read the mRNA. And it reads A, U, G, and it knows A goes with U, A, and C. So it will find an anticodon that matches this codon A, U, G and put them together. Now this little bit right here is called a tRNA. And a tRNA is a big piece of RNA that's all folded into a T shape, and it carries an amino acid. Now the amino acid that it carries is specific to the three nucleotides that are sticking out of the bottom of it. These three nucleotides are called an anticodon, and the anticodon UAC is always going to carry the amino acid methionine. We plug it back in. Then the next one would be GCG. which is arginine. Then the next one would be AAU, which is leucine. The next one would be CUU, which is glutamine. Then we have GGG, which is proline. UCC, arginine, and AUU, which is stop. So we now have all three kinds of RNA at work here. The rRNA, ribosomal RNA, is doing the job of matching up the codon to the anticodon. The mRNA is the copy of the code that was taken from the DNA. And finally, the tRNA, transfer RNA, brings in the amino acid. And by the way, mRNA, the M stands for messenger. So now that we've lined up the amino acids, we will recall that a protein is made out of a chain of amino acids. So these amino acids need to bond together, and they bond using a peptide bond. Once they bond, then this tRNA will go out and find another methionine and do the same thing all over again. This one will find another arginine. This one will find another leucine. My amino acids are out of control. There we go. Another glutamine. Proline. Peptide bond. All right, it's not the same peptide bond, but that's okay. Don't forgive me. You get that the rest of these are all bonded together. Yay, technical difficulties. We love it. There we go. Go find another arginine. And finally, the stop codon isn't actually an amino acid. It just, the RNA, when it reads the stop codon, it knows it's all done. And so we have our lovely chain of amino acids. This is known as a polypeptide. This will be folded and bent and combined with other chains to make the protein. So this is the kind of baseline structure of the protein, then it gets folded and bent and becomes a protein. And it goes and does its job. Meanwhile, we have this mRNA, right? It's still beautiful, it still works. So what it does is it runs through the rRNA a few more times to make more copies of that protein. And once it's all done, then the nucleotides, it gets broken down. The nucleotides go back to the nucleus and they get reused to make a whole different strand of mRNA later on when we need to make a different protein. And one last thing. Oh, yes. This process was called transcription. This process, where the rRNA reads the mRNA, uses the tRNAs to build the protein, is called translation. This entire thing, both transcription and translation together, are known as protein synthesis. And back in the nucleus, the reason we even have DNA nucleotides, because we didn't use them in this process. The DNA nucleotides are used for the process of RNA replication, when you have to double your DNA and make new DNA, like if you need to make some new cells. All right, that's it. I hope it helped.